then I'll make a start with section 2. What I have posted on the web as 2A, 2B is still part of the exam, I think. Alright, so we've looked at membranes. Membranes have lipids, membranes have proteins in them. The proteins give the action, the lipids give mainly the barrier function. So how do lipids form a barrier? Well, they're amplified molecules, and that takes care of their orientation, so you get a bilayer. But how does that form a good barrier? Well, the idea is that lipids have fast rotation around their vertical axis. Think about, can't help it, the figure skaters. Now when they're doing this fast pirouette, if they're going really, really fast, you don't even see who the person is anymore. It becomes a cloud. The same happens with these lipids. This is a very fast motion that they have. It's really just molecular motion. So you get this average shape of the lipid molecule, and put a bunch of those together, and you get something that forms a good barrier. And if some of you play the game of foosball or table soccer, and you know that you can have a very good defense if you take your goalkeeper and you let the goalkeeper go back forth very quickly, and that holds the opponent, that keeps the opponents from shooting the ball in your goal. And if this rapid motion of all these lipids vibrating, that actually helps this barrier function. Okay, now this shape is a cylinder, which is what we're getting, and the average cross section through the head group and the average cross section through the lipid part is about the same. You get about the same cross-sectional area here and there. And you get a cylindrical shape. But it can be different. It can be that the head group is relatively small or relatively large. So if the head group is relatively large, for instance, if the head group is more charged, if it's a serine that is esterified there, or an inositol. Inositol is a sugar, alcohol is a pretty big molecule. Then the head group is relatively large relative to the fatty acid part. And then you get this beer glass shape, I like to say. So now the head groups actually take up a larger volume. If we arrange these molecules together, you get something that's curved. On the other hand, if we have a very small head group, such as if we have ethanolamine as the alcohol esterified to phosphate, so phosphate to ethanolamine, you have a very small head group. You get this type of shape, the inverted cone. And you would perhaps be surprised, but the most abundant type of lipid that we find in biological membranes is not the one in the center, but it's the one on the left, and especially here on the right. In E. coli, 80% of the phospholipids are phosphatidylethanolamine. They look like this. So you think, how is that making sense for making a membrane? Well, membranes have proteins, and the proteins have to fit in, and the proteins are not straight cylindrical shapes. They have all kinds of irregular shapes, and so they need this type of lipids, the extremes, in order to make them fit in. That's one. And secondly, most membranes are highly curved. There's curvature. Think about here, a membrane in a curve. Then you need these guys with a small head group and a relatively large lipid volume to sit on the inside of the curve, and on the outside you need these beer glass shaped lipids. So the lipids that we have here on the left and on the right, if we have those isolated, purified, only by themselves, and we try to put them in water, they will not form a bilayer. Only the guys in the middle will really form a nice bilayer. But in biological membranes, the extremes actually dominate because together they allow the membrane to be curved and they allow the membrane to accommodate irregular shaped proteins. How's that? Does that make sense? Okay, so what else do we need to know about lipids and how they behave? Well, I've said that the model for the biological membrane is called the fluid mosaic model, where the proteins float around in the lipids. And that means that the lipids have to allow for a lot of motility. And the lipids themselves also move around. So the way this is depicted is we say the lipids are in this liquid phase. And what this really means is that the van der Waals interactions between the fatty acid tails of the separate lipids are weak. There's distance between the fatty acids of the individual different phospholipids. We can have a different situation if the temperature is low, lower than the normal temperature for that biological membrane, then we get van der Waals interactions that take over, and we get a close arrangement of all the fatty acids close together. And we call that the gel phase. So there's a figure in the book, and that depicts the same idea. Here we have the liquid phase, where the fatty acids are a little bit removed from each other. There's motion in those fatty acids. There's a lot of molecular motion. And in the gel phase, there still is molecular motion, but it's much less. And the van der Waals interactions between the fatty acids are strong. Now, this gel phase doesn't form a good barrier, but you actually need that dynamic property of the lipids, back and forth motion. You need that in order to form the barrier, just as your foosball players on your table soccer table. So how do we get above that point where the membrane is no longer in the gel phase? So we can do experiments, and if we take membrane-forming lipids and make membranes and put them in a machine that actually measures how much heat you have to put in order to reach a certain temperature. So if we start here in the gel phase and we increase the temperature, then we see that, or if we input heat, we see an increase in the temperature, but at some point we have to put in extra heat in order to get to a higher temperature. And that extra heat goes into the melting of these fatty acid chains here. So in this type of experiment, you can see that there is this transition from the gel phase to the liquid phase. So what makes that transition happens at a higher or at a certain temperature? Well, the melting point, it becomes lower if we have more unsaturation in the fatty acids. That is to say, more double bonds. And we need especially cis double bonds. Trans double bonds won't do it. Why is that? Because cis double bonds make a kink in the fatty acid. So that creates more distance. Shorter fatty acids have weaker van der Waals interactions between the fatty acids in the same lipid molecule. So that also makes that you get a more liquid state. And if you have a larger head group, you increase the distance between the fatty acids also, because the head groups force the lipids away from each other. So that also tells you again why not all the lipids in the membrane are the same. There's not just one membrane forming lipid, there are different kinds. You need an optimal mixture to have this nice, what we call liquid phase. And the liquid phase is important once again because in a lot of important biological processes, the proteins have to be able to float around in that membrane. Now, the sterols have a function in this process as well. It's thought that the sterols sit between the phospholipids with the polar head group, that's a single hydroxyl group, between those polar groups and the rings and that short acyl chain sticking in the hydrophobic layer. So there's once again the model that we see in our book. And now if we do that experiment where we take a membrane and we go at a low temperature where we know it's in the gel state, and we look how much heat we have to input in order to get a temperature increase, then we see that that amount of heat that we need in order to transition to the liquid phase is lower. And what that really tells us, this experiment, is that the presence of sterols, so the presence of cholesterol in your membranes, makes that the membrane has a liquid type behavior over a wider range of conditions. So, because the barrier function requires the liquid state of the membrane, you have a better barrier function with cholesterol. So cholesterol is a good thing. You need to have cholesterol. And you make cholesterol yourself. You can also eat cholesterol. What's bad is cholesterol esterified to a fatty acid. Those are cholesterol esters. Those are not so great to have because they can clog arteries. Alright, now let's look at the proteins. Look at the lipids, let's look at the proteins. There are three ways in which proteins can be associated, can be incorporated in the membrane system. First we have integral proteins depicted here. This just depicts two ways in which integral proteins can be integrated in the membrane. So integral proteins have part of the polypeptide sticking out on both ends, and they go across the hydrophobic layer. We know that we can that we have an integral protein if that protein can only be extracted from the membrane by using detergents or soap molecules. Those detergents associate with the protein, they replace the fatty acid parts of the lipids here and allow this protein to become solubilized. So they're only extracted with detergents. Second type of protein is what we call an anchored protein or lipid anchored protein. And there are different classes of these depicted in our picture here. Mainly on the inside of the plasma membrane, we have sometimes fatty acids that are attached to cysteines of proteins. Can be attached to other amino acids. Cysteine is one option. And that fatty acid inserts into the membrane hydrophobic layer, and in that way the protein is anchored. Some signaling proteins that we'll see later in the class have this type of anchoring. There are other options. Some proteins are attached to a special membrane-forming lipid which is called phosphatidylinositol. So then we have that inositol here, and then there's a couple of extra sugars which are modified attached, and to that attached is our protein. So we call this a GPI anchor, or glycosphatidylinositol anchor. And these types of attachments are found only on the outside of cells, and they have some medical importance. Some parasites, such as the parasite that causes the African sleeping disease, as well as malaria, they have these GPI anchored proteins at their surface, and what happens is these organisms change the protein that's on the surface all the time. So they have genes, hundreds of genes, that can be expressed, but only one or two is expressed at one given time, and they continuously switch the protein that they have on the surface, which makes it very difficult for the immune system to attack those cells. So the only way in which you can maybe deal with this type of invader is by having a system that interferes with the GPI anchor synthesis specifically in that organism. So there are some research groups who try to make drugs that prevent this type of anchoring in malaria and related parasites. Let's keep it at that. Okay, then the third way proteins can be associated with the membrane is what we call peripheral. And peripheral proteins are just associated by binding to the lipid head groups by means of salt bridges, so ionic interactions. And these can be released with high salt or with going to extreme pH like pH 11, very basic condition. You tend to remove proteins from the lipids. The anchored proteins are removed by detergents or by enzymatic cleavage of the anchor. So there's three ways proteins can be associated with the membrane. We are most interested in the integral proteins. So if we think about the integral proteins, we can ask the question, what is their possible function? And there are many functions. One is to be a signaling molecule that communicates with the outside of the cell, and you're a receptor. We have receptors on the surface of our cells to which hormones can bind, and in this way, cells of the body can communicate with the rest of the body. They can be anchors keeping the cell in a certain place, so attaching it to extracellular matrix or something on the outside of the cell so you organize cells. Importantly, some are transporters. They allow molecules to get in or to get out of the cell. Some are channels, a special case of transporters we can say, but channels don't bind the substrate. Channels allow for very rapid transport. Important in the nervous system, we have sodium and potassium channels that play crucial roles there. Some of the transmembrane proteins are required for fusion processes, and this is something that viruses do. Viruses need to often fuse their envelope with the membrane of the host cell in order to get in, so fusion proteins. And then a lot of integral proteins are enzymes. They function in the synthesis of lipids or other processes that have to happen at the surface of the cell. So here we have a depiction of the integral membrane protein. And we have here, say, in extracellular space, this protein might have sugar groups, might be glycosylated. Then it crosses the membrane, and then we have an intracellular part, or intracellular domain. And when the protein crosses the membrane, it can be as an alpha helix. And the membrane is 3 nanometers in thickness. So 30 angstroms, 3 nanometers. That's the same. You might remember angstrom is often used as a measure of distance. It's 10 to the negative 10 meters. And it's approximately an atomic distance. That's the angstrom. So 30 angstroms, 3 nanometers is the approximate distance between the hydrophilic boundaries on both sides of the bilayer. So if we make an alpha helix and we know what the alpha helix looks like, we can count the approximate number of amino acids that we need. And it's 18 to 20 amino acids is what you need in order to cross a membrane as an alpha helix. So you can ask the question, why an alpha helix? There are other options, but why is the alpha helix important for integral proteins in many cases? And the answer is that in the alpha helix there's hydrogen bonding between hydrophilic groups of the amino acid polypeptide backbone. So the carboxyl group and the amino groups of all of these amino acids are satisfying their hydrogen bonding needs with each other. They don't have to do that with water. And then the side chains, all these R groups of these amino acids, are hydrophobic. So this is an arrangement that is happy in this bilayer. So we can talk about this with a different diagram. And what it comes down to is we have 3 possible arrangements by which a protein can cross a membrane. There's 3 possibilities. So the first one I just talked about is an alpha helix. It will take 18 to 20 amino acids. The amino acids have to be hydrophobic with their side chains. Something like a lysine or an arginine or a glutamic acid would not be happy within the hydrophobic area of the membrane because it wants to associate with water. So we need to have apolar or non-polar side chains, and the side chains all stick out to the outside of the helix. If the helix goes like this, and the side chains are all to the outside. And from top to bottom, all the amino acids with their backbone are hydrogen bonded with other amino acids in the helix. So there's another option, because you can have a couple of transmembrane domains coming together, and actually 6, 8, 10, and 12 is often seen. But you can also have 1, 2, or 3. In this case, you can have a slightly different arrangement. You still have an alpha helix, and any part that's in contact with the fatty acids is still going to be non-polar or apolar, or hydrophobic. But where the helices, where the protein parts make contact, you can actually have hydrophilic amino acids coming together. So you can imagine here in this set of helices here, they can have contact points where we have hydrophilic amino acids. And now you can imagine that if we have over 6 or more of these hydrophilic amino acids sequences in alpha helices, that we might actually form a structure where everything that's in contact with the fatty acids is hydrophobic, but on the inside we might have a hydrophilic surface which can form a channel, or salts, or ions. Okay, so we would have polar contacts between the alpha helices. It still takes 18 to 20 amino acids. So there's yet another possibility, because you can also cross the membrane as a set of beta strands. But then you need a whole lot of beta strands together. You need just 7 to 8 residues to span the membrane, because the beta strand is much more extended than the alpha helix. It takes us 7 to 8 amino acids. But in this case, you need the amino acids that are in contact with the fatty acids again to be hydrophobic, and the ones that are in the center, they can be polar. And this type of arrangement is commonly found in the outer membranes of bacteria and in the outer membranes of mitochondria and of chloroplasts. Whereas this type of arrangement with one or more alpha helical structures is common in all other situations. Alright, so I'd like you to understand the 3 possible arrangements that we can have for crossing a membrane as a protein. It is a favorite exam question. Okay. Now, I've said that you have the fluid mosaic model. What that means is the proteins move around in the membrane. So, I'm just saying that. Why should you believe me? Well, there are experiments that people have done. The following is a description of one type of experiment that shows the mobility of proteins in the membrane. So if we have cells from 2 different organisms, then the cells will have different proteins in their membranes. And the different proteins can be followed by using antibodies that recognize those surface proteins. So what if we fuse those 2 membranes? If we use a trick and we fuse the 2 sets of cells, we initially get domains where the membrane originated from this one cell, say the mouse. Most of that membrane is still separated from the membrane of that human cell. But if we wait some time, then everything mixes. And we can follow the presence of the proteins by using immune fluorescence. So you come with an antibody. The antibody is bound to a fluorescent group. Let's say a green fluorescent antibody recognizes the mouse proteins, a red fluorescent antibody recognizes the human cell proteins. If you take that picture, you see in the immune fluorescence, I'm going to put you in a slight dark situation here. We see here clearly there's a red and a green zone on this cell. This is a living, not a living cell, it's fixed, right? Because we've given it an antibody. But if we do the same experiment after 40 minutes, we see that everywhere the green and the red is mixed. So the proteins have mixed. But it indicates that the proteins of the two cells are moving around. They're floating freely within that lipid sea. Now, the only proteins that do not float around freely are those that are attached to the cytoskeleton, that's the structure within the cell, or the proteins that are attached to extracellular proteins, which are called extracellular matrix. So not everybody moves around freely, but most proteins do. Okay, now we get to a conceptually interesting, but I think very important topic in the class. It's not terribly well treated within the present book, so I'm using some diagrams that I got from elsewhere. If we look at a eukaryotic cell, it's a highly underappreciated but important point, we can see that for every membrane, one side of that membrane is facing the cytosol, or it's facing a compartment that used to be the cytosol. So let's go here. We're on the outside of the cell. Here we have the membrane. These two lines represent the bilayer. We cross the cell, now we're in the cytoplasm, in the cytosol. Here in this whitish, grayish area, and in this pinkish area we're on the outside. The membrane faces these two sides. Now we go out of the cytosol and into the lysosome. We do the same thing. We're here on one side, we are facing the cytosol, on the other side, we're facing the lumen of the lysosome, which is similar from the perspective of the cell to the outside world. And if we go to the Golgi and the ER and the nuclear envelope, it's the same thing. Now, what happens if we go into the mitochondrion? The mitochondrion has two membranes. We cross the outer membrane, we go from the cytoplasm into what is sort of an outside world, the space between the two mitochondrial membranes. Then we cross the inner membrane and we get into the mitochondrial matrix. Now, in an evolutionary sense, the mitochondrion is derived from a bacterial ancestor. So that bacterium had a cytoplasm, and that matrix is still equivalent to the cytoplasm of that bacterium. So we're once again in a cytoplasmic-like environment. This rule is always true. There's only one exception, and this is the outer membrane of gram-negative bacteria. There are two major groups of bacteria. If you think of the blue bacteria, gram-positive have only one membrane, and gram-negative have two membranes. The gram-negative bacterial outer membrane really faces two outside-like compartments. Nothing faces the cytosol. But this is a rule that's always valid in the eukaryotic cell. There's no exception that I know of. It would rock my world to find the exception. So we can say every membrane faces two distinct types of solutions. On one side, you're always in contact with what's cytoplasmic-like or cytosolic-like. So the real compartments that we're talking about are the cytosol, the nucleus, the inside of the nucleus, which is connected by big pores with the cytosol, the matrix of the mitochondrion, and then in plants, also the stroma of the chloroplasts. That's also an ancient cytosolic compartment. If we think back of the ancestor of the present chloroplast, it was a cyanobacterium, and it had a cytosol. So for the outer side, what do we have? So we have the outside of the cell, but also the lumen of the whole endomembrane system. So there's the lumen of the ER, the space in the Golgi, the space in the lysosome, and the space in the vacuole. The peroxisome is another compartment that is distinct from the cytoplasm. The space between the mitochondrial inner membrane and outer membrane, we call it the intermembrane space. And then in chloroplasts, the space between the two membranes of the envelope, that is the outer and the inner membrane, and then the space within the filoparts, that is the lumen. And then finally, the periplasmic space in bacteria qualifies as the same. Now I'm telling you, these two compartments are fundamentally different. So there's, again, one exception, the outer membrane of gram-negative bacteria. It faces two compartments that are both to be considered as exoplasmics, outside of the cytosol. So how are these two compartments different? Well, think about what we find on the inside of a cell in the cytosol. And we typically have not typically, always have low concentrations of sodium, very low concentrations of calcium ions, and relatively low concentrations of chlorine ions. We have abundant potassium. Potassium is the dominant ion within living cells. Let's not forget that. So that's always high. The pH tends to be pretty neutral. If you think about what's on the outside, then we have always a higher sodium concentration, relatively higher, but still fairly low calcium, and relatively high chlorine concentration. The pH can be extreme, is often low, but not always. So we often have a higher proton concentration, but that's not always the case. Really, if you think about the evolution of cells, this makes sense. Cells, this is really my personal belief, opinion. I'm pretty certain I'm right in this. What I'm thinking is cells evolved like 3.5 billion years ago. And if we look at the chemistry in the cytosol of cells nowadays, they're all very much the same. And they're all pretty high in potassium and low in sodium. Over the time, on our planet, what happened is the sea became sulfur. Life evolved in the sea. And so cells wanted to hang on to the chemistry in which they originally evolved, and they had to keep sodium chloride on the outside. That's just what they did. Okay, what are the differences do we have? Within the cell, it is typically a reducing environment, which is also something I think that cells have taken from their early evolutionary steps. What do I mean that there's a reducing environment? What I mean is there are coenzymes and small molecules that can donate hydrogen easily. And these molecules